Today let us learn about complement pathway. So from where the complements are formed, first of all we will discuss about the origin of complements. So you know that there are mainly 9 complements, C1, C2, C3, like that up to C9. And in C1 itself you have C1Q, C1R and C1S. And which complement is present in maximum concentration? That is C3. And the minimum concentration you have C2. So from where these complements are formed? They are formed from several organs. Okay, so C1 is formed from GIT, that means intestine, and C2 is formed, C2 and C4 from macrophage, C3, C6, C9 is from liver, C5 and C8 is from spleen. So from these the complements are formed and they are present in our blood. They are present in the sera. So they are actually non-specific proteins which are present in serum. That means normally they are inactive state and certain factors will activate them. That means normally they will not bind to free antigen or they will not bind to free antibody. But in some cases, for example, if there is any inflammation, in inflammation and all they will be increased. Okay, that is called acute phase proteins. That means such proteins which increase in inflammation during inflammation is called as acute phase proteins. So this complement proteins are one among them. Okay, so these complements they will increase during inflammation. That means when there is an antigen antibody complex, the complement will be activated. There is a free antigen or a free antibody, they will not be activated. But for an antigen antibody complex will activate these complements. And also some other factors are also there. So once the complements are activated, they can go through two pathways. One is the classical pathway, other one is the alternate complement pathway. So these two pathways, whether they go through both, the final result is the same. That means the lysis, the damage or the destruction of the target cell. So these pathways, which pathway they are going, that means what determines this? If it is an antigen antibody complex, they will be activated through classical pathway. If it is some other like lipopolysaccharides of some cell wall or some zymosins, nephritic factors, in such cases they will be endotoxins, mainly endotoxins. So these will activate them through alternate complement pathway. And you have some other pathway, that is lectin pathway is also there. Mainly you have to remember these two, classical and alternate complement pathway. So the result is same. Now we will discuss in detail about each pathways. First is classical pathway. Classical pathway, as I have told, it will be triggered by antigen antibody complexes and the first complement which is activated. So here the antigen antibody complex, antibody is involved here, and this complement when it is activated, to where it will bind, to the antigen or to the antibody? To the antibody, the Fc portion of the antibody they will attach. Okay, and which complement is activated first? In classical pathway, C1 is activated first. In C1 itself you have 3 subunits, C1Q, C1R and C1S. You can see the microscopic picture of C1. So this C1, initially the C1Q will be activated, then it will activate C1R, then it will activate C1S, and this C1S will in turn activate C4, the complement number 4, so that it is split into C4A and C4B. So here the C4A will go out. So now you have C4B in the pathway, which activates C2 into C2A and C2B. Here the C2B will go out, so you have C2A in the pathway. Just revise C1, C4B, C2A. Together called as C3 convertase, which in turn converts C3 into C3A and C3B. So what happens? Who will go out? C3A or C3B? C3A will go out, so you have C3B. Now this is called, revise, C1, C4B, C2A, C3B together called as C5 convertase. Convert C5 into C5A and C5B. This C5B will activate C6, C7, C8 and C9 to form membrane attack complex. So that is about classical pathway. Just revise C1, C4B, C2A, C3B, C5B, C6, C7, C8, C9. Now we will discuss about alternate complement pathway. Here these are triggered by endotoxins, zymosins, nephritic factors, lipopolysaccharides of cell wall, etc. And these here the antibodies are not involved. It's not antigen antibody complex. So initially it is beginning from C3. That means you don't want C3 convertase here because already these factors will stimulate C3 and break it into C3A and C3B. So who will go out? C3A will go out. Now you have C3B. C3B will bind with a factor B and splits this factor B into factor BA and factor BB. So you have C3B factor BB, that is C3BBB, and this is called as C5 convertase, which converts C5 into C5A and C5B. This recall C3BBB and C5B. Now you have C5B activates C6, C7, C8, C9 to form membrane attack complex. Okay, so that's all about alternate pathway. C3B, BB, then C5B, C6, C7, C8 and C9. Lectin pathway is actually beginning by mannose binding lectin. Mannose binding lectin will bind to the carbohydrate which is present on the cell wall of the antigen. So here also antibody is not involved, and the first complement to be activated is C4. So in the classical pathway it was C1, in the alternate pathway it was C3, and in this lectin pathway it is C4, and the remaining is similar to that of the classical pathway. So the initial thing is mannose binding lectin binding to the carbohydrate which is present on the cell wall of antigen. Now you have, this is the microscopic picture of the membrane attack complex. You can see here that's formed by the C5B will activate C6, C7, C8, C9 to form membrane attack complex. Now this is the summary of the complement system. So at last what happens to the cell? The cell sometimes they will insert perforins. So what happens? There will be so many pores present in the cell membrane, so that the contents of the cell are released out and at last the cell will die. Okay, the integrity will be lost here, and in other cases they will attract some mast cells and they will undergo degranulation, or sometimes they attract the neutrophils or some other phagocytic cells, and these cells undergo opsonization. Phagocytic cells, they will phagocytose these target cells, and to make the opsonization, you know, C3B is the C3A and C3B. In that, the component C3B is the best opsonin. It will make the target cell tastier, so that the phagocytes will feel it very tastier with the presence of C3B. So in that way, opsonization can also occur, and during these times C4A and C5A are going out of the pathway, but you should not think they are not significant, because they are some anaphylactic molecules, or they are chemotactic. So they can cause some changes, increasing vascular permeability, attracting some chemokines, etc. So these all happens. And in some conditions, some autoimmune diseases like SLE, rheumatoid arthritis, due to this excessive activation of these complements, the free complements available in the blood will be less in these conditions, SLE and rheumatoid arthritis. And in some cases, like in gram negative septicemia or dengue hemorrhagic syndrome, in that cases and all, the complements are activated and they are adhering to the platelets, so they will lead to thrombocytopenia, and there will be disseminated intravascular coagulation. So these all are the significance of the complement system. So there are certain conditions where there is deficiency, certain diseases where there is deficiency of this complement proteins. Okay, so you can remember it like her sister returned back. That means her, that is actually hereditary angioneurotic edema, that occurs when there is a deficiency of C1 inhibitor. Remember, it's not C1, it's deficiency of C1 inhibitor. Now next is her sister. So sister, that is SLE, systemic lupus erythematosus, occurs when there is a deficiency of C1, C2, C4. Now the third one, her sister return. So that means recurrent pyogenic infections occurs when there is deficiency of complement protein C3. Now her sister returned back. So bacteremia, that will be there when there is deficiency of C5, C6, C7, C8 and C9. So that is very easy to remember, and that's all about the complement system.